Hello, and welcome to this webinar, Quality Drug Candidates in Under 2 Years. My name is Rhys Armstrong. I'm the editor for DDW, and I'm pleased to be introducing this event, which will explore how Evotec is planning to shorten timeline for small molecule drug discovery. Shortly, we'll hear from a panel of Evotec speakers who will explore the current challenges facing small molecule drug discovery and how Evotec's every compound a candidate approach can accelerate drug discovery while improving quality. There'll be a Q&A after the panel, so please drop your questions into the chat and we'll get them to the speakers. Okay, with that, I'm pleased to hand over to Ashley Jarvis, EVP and Head of Global Chemistry at Evotec. Ashley, over to you.
Okay, thanks Rhys and DDW, and welcome again everyone to today's webinar. And the topic is Evotec's Every Compound a Candidate, or ECC strategy. I'm delighted to be joined by the authors of a recent Drug Discovery Today publication that describes this approach at Evotec, and we're going to discuss some of the features of the strategy as well as an opening up to questions from the audience. But before we get into it, let's start off with some brief introductions. So I'll go first. So my name is Ashley Jarvis. I'm the head of global chemistry at Evotec. I've been with Evotec for over 8 years now, and I have a real passion for integrated drug discovery, and I'm a really strong believer in what we're trying to do with every compound a candidate. So my background is predominantly in small molecule medicinal chemistry, but I have had the chance to work in different modality areas applied to a number of therapeutic areas and target classes. I'll just keep it brief there for now and bring in Dermot.
Yeah, thanks, Ash. Good morning, good afternoon, everyone. My name's Dermot McGinnity. I'm the head of Global Discovery DMPK here at Evotec. I've been at Evotec just over 2 years. I joined from AstraZeneca, where I was the head of DMPK within the oncology R&D unit at AZ. And I was at AZ for over 25 years at a range of different roles, all within DMPK, discovery and development across different therapeutic areas. So delighted to be here today and tell you more about every compound a candidate strategy. And I will pass you over to Jerome.
Yes, hi everyone, I'm Jerome Bénérolle, so medicinal chemist by training, and spent 20 years or more than 20 years in pharma industry, leading projects from ITID to candidate nomination. And a few years ago, I decided to join the molecular architect department to combine AI, CompChem and MedChem expertise to progress project more rapidly. I'm also leading the DMTA performance group in Evotec, and today I will focus my talk on the overall process we pursue during this analysis of our portfolio. So happy to be there and discuss with you all, and I pass then to Christophe.
Yeah, thanks, Jerome. So I'm Christophe Bollon, I'm a medicinal chemist by training, and I've been spending a bit more than 20 years in the pharma industry, first 10 years in Sanofi, and since now 10 years at Evotec, where we are supporting the entire pharma world for their drug discovery projects. So I'm very glad to be here with you today because we are going to discuss 2 main papers that we have published in 2025 around every compound candidate first, but also we'll try to discuss a bit the importance of AI/ML and how at Evotec we manage to run AI/ML within our drug discovery projects, accelerating all the process. So very glad to be here today and start the discussion.
Okay, so thanks, guys. So before we get into it, I'll just remind the audience about the format. So I'll be sharing an initial discussion with the authors, with the team, and then we'll move to audience Q&A. So please get those questions into the chat, and then we'll get to them at the end. So let's get the conversation started. And Dermot, I'll begin with you. So can you briefly summarize what ECC is?
Okay, so ECC, every compound a candidate. So at a high level, Ash, it's a strategy, it's a set of tactics focused on improving both the design and profiling phases of small molecule drug discovery. That's what it is. It focuses on the speed and the quality of candidate drug delivery. And essentially, and again, at a high level, and we'll go into all the details, but we advocate for combining operational excellence, firstly, with more experience-led risk-taking in the design and profiling phases, secondly, and thirdly, and importantly, we want to increase our translational predictability. Okay, so what does that mean? For example, one tactic is to progress more compounds through downstream, both in vitro and in vivo models. And what we want to do here is reveal what we call translational thresholds, or indeed critical blockers for compound progression. So ultimately, in order to deliver higher quality candidate drugs rapidly. And we'll expand on all of these different elements during the webinar. So that's what every compound candidate is.
Okay, that sounds good. Sounds compelling. So let's build on that. So how did this strategy emerge, and maybe the name? Why is it called every compound candidate?
Okay, so I'll come to the name in a minute. So yeah, it was a combination of all the authors bringing their own ideas and perspectives, not just from Evotec, but also from companies we've worked from previously in the past. But also, what we wanted to do is one of the first conversations that we had around this was asking a central question: how do drug discovery teams within Evotec, but also beyond, because we think here this has every compound candidate has lessons that can be learned outside of Evotec. But ultimately, how do drug discovery teams within Evotec reduce timelines whilst increasing quality of drug candidates? That was the central question that we posed. But rather than just come up with ideas, we needed to provide a data set and a framework around those ideas. So what you'll hear from Jerome in a few minutes is we evaluated our partnered small molecule portfolio delivery over the recent past. And you'll see some data around that. But also then we had discussions with project team leaders, with key stakeholders within Evotec, in order to identify both enablers, but also blockers towards rapid quality candidate drug nomination. And so we'll share that data analysis, and we'll share a distillation of all those ideas and suggestions into a set of tactics and a strategy. So that's how it evolved. With respect to the name, I guess the first thing to say is clearly not every compound will, or indeed should be, a candidate drug. But this framing underpins an organizational culture that reflects every compound being appropriately evaluated and highlights what we call the attentiveness. And we'll discuss this in more detail. The attentiveness required to identify and rapidly progress potential candidates. Ultimately, it captures the overall urgency of the mission, to bring clinically meaningful drug candidates through the process towards nomination and IND submission. So we like it.
Yeah, thanks. I think that'll really help the audience to understand that it's that attentive mindset, obviously with a framework, not saying, right, every compound's going to be a candidate. I think it'll probably be a question that'll come up later. So perhaps an obvious next question then, maybe framing the problem statement. So is there an actual problem with the speed and quality of candidate drugs that needs to be solved?
Yeah, good question, Ash. And it's a problem not just for our organization, it's a problem for the industry. And frankly, it's a perennial problem. So I joined the industry in the late 1990s, and increasing the productivity and the efficiency of pharmaceutical R&D was a problem then, and it remains a problem. And there are also now several macroeconomic competitive pressures, increasing competitive pressures, currently affecting drug discovery organizations. Yeah, so we know the cost of new therapies is an increasing economic burden for health systems worldwide. But actually, if we just focus on small molecule drug discovery, we know there's a real cost of failure of innovation with, and again, everyone can choose the number, but it's a high percentage, up to 90% of candidate drugs fail in clinical studies. Okay, there are a number of different reports out there, but that's a real problem. It's a real problem that we have to solve. So there's a competitive pressure to be quicker and to be more efficient and to reduce that clinical attrition rate. And of course, if you just take a step back from all of those sort of facts and figures, ultimately there's a profound and urgent need to discover and develop innovative therapies for patients. So it's not just true of our organization, frankly, it's true of all drug discovery organizations, that we have to employ optimal strategies for the discovery and the development of small molecule candidate drugs. We have to do it more rapidly, more cost effectively than historically realized, and we have to have a higher probability of successful approval for compounds that deliver, significant, meaningful clinical benefit. So it's a problem that we share with all drug discovery organizations, and we continually need to improve. This is on the background as well. One thing that has changed, since I joined the industry, is this is on the background of increased expectations of small molecule drug discovery evolving, towards more challenging drug targets and also more challenging modes of action. So, we know we've got targeted protein degradation is a key mode of action in the last decade or so. Likewise, covalent inhibitors, targeting protein-protein interactions, targeting RNA, et cetera. So the bar is being raised whilst the overall competitive pressures to be quicker and to improve quality remains. So there's a problem to be solved, absolutely.
No, thanks, Tim. I mean, that certainly resonates with me. And as you say, we're trying to navigate the benefits of operational efficiency, but we're navigating ever more complicated biological mechanisms and different modes of intervention, and all that's got to be brought together. And let's do that. So you mentioned data. So let's bring in our expert for data analysis, Jerome. So Jerome, can you tell us a little bit more about the data analysis that underpins and supports ECC, please?
Yes, sure. Of course, the statement that drug discovery is too slow and too expensive is supported by data from literature. And we have some of those data which put average cycle time from validated hit to nomination of the candidate ready to enter this IND enabling phase in the pharmaceutical industry at 4 years. 4 years is a median value for more than 200 data points and from 19 companies selected between 2018 and 2022. Interestingly, Evotec is outperforming the pharma industry by almost 30%, with an average timeline for this identification at 2.9 years, combining data from a selection of small molecules, 15 recent projects of Evotec. But our inspirational goal is to deliver the candidate in less than 2 years. By combining a continuous improvement on the foundation of deep disease expertise with technological differentiators, including AI/ML tools. But let's dive deeper into this analysis on the next slide, please. So we conduct this thorough analysis of our candidates portfolio with a particular interest of 15 projects, recent projects, small molecule recent projects. These projects share a common starting and end points, but yet they are also highly diverse in terms of indication, going from CNS project to oncology or immunology project. Our recent portfolio demonstrates also speed and quality. Quality because we deliver this in 2.9 years, but also 80% of this portfolio is still progressing through IND enabling to clinical phases. Instead of the conventional approach to project progress, we have proposed the division of the project into 2 distinct phases: the candidate design phase and the candidate profiling phase. Candidate design starts from the start of the synthesis to the first candidate synthesis, and the candidate profiling is from the candidate first synthesis to the candidate nomination. Okay. First phase demonstrates our capacity to generate optimal designs. The second demonstrates our efficiency in profiling those candidates, especially in terms of early tox studies that are time consuming, as you know. The variability observed during those two phases and across projects has led to informal interview of senior project leader or key stakeholders of project within Evotec. Those interviews being conducted retrospectively to evaluate, as mentioned by Dermot, key enablers of the project and key blockers of project delivery as part of also the lesson learned exercise we are performing in the company. Is on the number of compounds to achieve PDC nominations. The box plot below illustrates the number of compounds synthesized during project design phase with a median number of compounds of 577, and during the candidate profiling phase also with 251 compounds during this phase. The variability observed both in terms of timelines on the right-hand side of global timelines, but also number of compounds synthesized per project on the different phase, make us believe that optimization can be achieved in these two phases. The higher variability even on the profiling phase, when we discuss with a key stakeholder, appears to be highly driven by the importance of the attention to details and swiftly advancing promising compound candidate nomination. This is what we are calling attentiveness to candidates. And this was also one of the key drivers described earlier by Dermot, as every compound should be considered as potential candidates.
Okay, thanks, Jerome. Thanks for that comprehensive walk through some of the data. And you mentioned sort of conducting interviews and discussing with project teams and key stakeholders, which of course is a valuable source of information. So Christophe, bringing you in, because of course you were involved in that, can you describe what emerged as sort of the key areas of focus from those discussions with those groups?
Yes, sure, Ash. My pleasure. And I'd like also to thank the other colleagues for the very, very nice introduction. And I will try to develop some concepts that came out of these discussions. So discussions with these seasoned drug hunters at Evotec, but also our technological experts, led to some lessons learned and brainstorming sessions. And the main, even though those discussions been very similar, and with this goal of routinely achieving the timeline of less than 2 years from start of chemistry to the pre-development candidate nomination, we came up with these three main pillars that you are here depicted on the slides. So to the left, you have the first pillar, which is translational predictability. So with the development of modern dry and wet methods for biomarker detection, disease modeling, prediction-led decision-making, etc., we now have all the infrastructure to control this translational chain, which is so important to success. So all we do in small molecule drug discovery is really about predicting from in silico to in vitro, from in vitro to in vivo efficacy and safety, and ultimately predicting the behavior of a given compound in the human context, including the human dose, which is extremely important to understand as early as possible in the project, and the therapeutic index. So this chain of prediction from in silico to in vivo is at the heart of every compound candidate strategy, so that the project team can determine what we call the translational thresholds already mentioned by Dermot, and also identify the potential critical blockers early on in the course of the project. So translational threshold is probably a word that you may not have heard before. It's somehow a new term that we present today. So let me give you a brief example. This example is a local unbound drug concentration, which is necessary to modulate the targeted pathological pathway in a relevant animal model. This information is absolutely crucial and central to every compound candidate strategy, because once you know the minimal targeted concentration to modulate the pathological pathway, you can then calibrate in return all your in silico, in vitro, in vivo learning cascade, which will make your life easier for the following part of the project. And to collect this information, which is absolutely key, you don't need to wait for the perfect molecule, an early prototype compound with some manageable liabilities can make the job to test the hypothesis. So this was the pillar one, identifying the translational threshold and the critical blocker early on in the project. Pillar two is operational efficiency. Excellence in molecular design is, of course, essential to a rapid and successful preclinical candidate design phase, and providing quick answer to a given design question with rapid synthesis testing is also key to achieve our high objectives. We'll develop this concept a bit later in the presentation. So this pillar two is really about design, make, and test excellence and efficiency. Pillar three, to the right of the slide, is project decision making. From our retrospective analysis, it appeared that to make the right decisions, you need seasoned drug hunters equipped with AI/ML tools, clear roles and responsibilities, and the cohesion within the multidisciplinary team. This allows a strong focus on project objectives, elimination of unnecessary tasks, and efficient problem solving. Thus, you will save time and money. One very important aspect which was raised and already mentioned by Dermot and Jerome, and I will repeat it again because it's extremely key, is the team attentiveness to detect the potential candidate drug at the moment it arises. Previous publications from many different companies, but also our own experience over many years within different organizations suggest that quality compounds, including the future candidate, are made early in a project. But the project team continues optimizing without necessarily reducing the risk and adding the quality to the project. So this is a waste of time and money, which can be significantly reduced or even eliminated with an efficient learning cascade and the support of project dashboards and AI/ML-driven predictive tools. As mentioned by Dermot, not every compound will be a candidate, but if you come with the mindset that every compound you design, every compound you synthesize, every compound you test might be the expected candidate, then this may change the game. So to support this mindset, you need to advance a higher proportion of compounds into downstream assays and models to maximize the learning, again, the translational predictability, which will naturally translate into less time and money invested as the project progresses in the next phase of the project. And finally, the last concept we defined was a concept coming from the very known DMTA cycle, and we propose to replace it with a more contemporary approach, which we call D2MTL, and we'll come to that a bit later on.
Okay, thanks, Christophe. So for the audience, that there's sort of three features. We have translational predictability, we have operational efficiency, and we have the decision making on projects. So that's the pillars that you referred to. Thank you. So we haven't discussed, unless I missed it, AI/ML. I can see it hinted on the slides. But given its centrality to design, drug design and discovery, how is AI/ML contributing to ECC? And as I'm seeing here, I'm aware that there has been a recent perspective publication by Evotec authors that's likely to be relevant. So AI/ML, Christophe, and ECC.
Yeah, thanks, Ash. Yeah, the role of AI and ML is an excellent question, which of course we asked ourselves. So with the emergence of AI design tools combined with increasing impact of physics-based methods, lower HPC costs, this has led most of the pharma industry to explore different ways of working. So at Evotec, AI/ML is very important and plays a central and fundamental role. But our pragmatic position, which was recently published in this journal, which is here on the screen, in Journal of Medicinal Chemistry earlier in 2025, is that they are not currently sufficient on their own to deliver the targeted transformational benefits required in drug discovery. So in summary, AI/ML is key, but needs to evolve in the favorable human and process-driven context brought by the every compound candidate strategy. So at Evotec, we've invested in AI/ML through our AI/ML research and development group, which is called In Silico R&D. This group is in charge of adapting, integrating cutting-edge techniques into our technological stack. And we have also set an operational group, which is called Molecular Architects, composed of seasoned medicinal and computational chemists who apply these cutting-edge techniques to discovery projects. So this is not the main focus of today's discussion, but I invite you to read this GMedChem paper where we clearly define and explain how we approach in silico tools in general, but including AI ML to progress our project more efficiently and navigate the chemical space in an efficient manner. So this intense reflection around every compound a candidate led us to the creation of this D2ML concept that we already mentioned several times in the presentation. So D2MTL stands for Design, Decide, Make, Test, and Learn, by opposition with the DMTA, Design, Make, Test, and Analyze. And it was introduced by molecular architects as an evolution of this DMTA concept. So with the emergence of generative design and directed compound enumeration, there is much larger number of designs that can be generated at each cycle. This renders the selection of the process more and more challenging. So what we think is that this requires a very well-established and structured decision-making process. Hence, this we included within the DMTA a second D, the Decide, which emphasized the importance of the selection phase with the integration of active learning, when molecular designs generated by a combination of human and machines are prioritized. So we propose that establishing an efficient Decide phase in the workflow is key to achieving excellence in molecular design and ensuring smooth project progression. So this collegial exercise not only gives a structure to the drug optimization process, but very importantly, it also contributes to our teams building a deeper mutual understanding of medicinal and computational chemistry. The replacement of analyze with learn is quite obvious. It denotes the importance for both the human and the machine to learn, getting as much as possible out of the data generated, as well as the evaluation and retraining of the predictive models. So within the D2MTL, commercial softwares and in-house tools at Evotec's HPC data center are orchestrated through our molecular architects Python pipelining tool, the so-called MAPL, to support the design and decide, whose last step here is a collegial decision-making meeting to finalize the selection of information which compounds to push into synthesis. MAPL brings the tools and the humans together to support our excellence in molecular design. So this is a very, very important notion that accompanies this emergence of those AI/ML tools. We need really to have a structured computational pipeline, computational workflow. And on the next slide, we here present a representation which, to the right, which illustrates the impact of the every compound candidate strategy as a result of all the concepts we've developed and discussed today. So the x-axis represents the time spent in the drug discovery project or the number of compounds synthesized. So the identification of preclinical candidates, so the first synthesis of this candidate is a result of a succession of optimization cycles. The impact of good decision-making, efficient problem-solving, and high operational efficiency leads to the natural decrease of the turnaround time of each single D2MTL cycle. On the other dimension, the y-axis represents the design information collected per cycle, and over the course of the project, of course. So the more efficient collection of design information generated during each cycle, thanks to an optimal decide and design workflow, allows navigating the chemical space more efficiently and hence reducing the number of compounds and D2MTL cycles required to achieve the identification of the preclinical development candidates within short timelines as illustrated here. So you can see here the impact of the every compound candidate strategy. So finally, here is a representation of our learning cascade. This schematic sets a 5 days turnaround time that is achieved in our organization for tier 1 assays, which typically include biochemical assays against the primary targets, a minimal number of ADMET and physicochemical assays that assess lipophilicity, aqueous solubility, metabolic stability, membrane permeability, and again, against defined anti-targets. So for compounds that pass the minimum tier 1 data quality thresholds, tier 2 assays can include cellular-based and additional ADMET assays, and the first rodent pharmacokinetic profile, which is central to the every compound candidate strategy. Again, our organization is very, very attentive to the turnaround time, and we set a goal of 25 days, starting from the decision to make, so meaning the end of the design phase, the time where you decide to synthesize a compound, to the completion of the chemistry of the synthesis, the tier 1 assays, upon generating the data package, including rodent PK data. So 25 days from start of chemistry to generation of the PK data package. So this is the typical minimum data package to assess the quality of the new chemical entity and whether that compound should be rapidly progressed into further downstream studies, such as the quick exploration of PK/PD or higher species PK. The rapid synthesis and collation of crucial early safety and efficacy datasets is recognized by our organization as being crucial for the rapid progression of quality compounds, thus leading to a clear definition of the translational threshold, the accurate refinement of human PK dose and therapeutic index, and ultimately the rapid and informed nomination of high quality candidates.
Dermot, can you maybe just sort of build and help the audience understand how screening, profiling sort of works in the ECC strategy compared to the perhaps the sort of conventional funnel type that we're all familiar with in small molecule drug discovery?
Yeah, thanks, Ash. Yeah, it's very nicely explained by Christoph what the tactics are and what the real life implementation is of ECC, at least one element within the D2MTL cycle. But one feedback that we got from when discussing this concept is, well, this is obvious, guys. Come on, what's, you know, what would you do differently? Isn't this what everyone does? Well, no, actually, it isn't. And we tried to describe it in this slide to really, it might be helpful for the audience to really fully understand the mindset of ECC as well as the practical implementation and how to compare and in this slide contrast against sort of an alternative way. So we really do aim to advance a high proportion of compounds in downstream assays and models, yeah, in order to maximize learning. That's the first thing to say. That's different from maybe what we've done in the past and maybe what other organizations do. And we should recognize perhaps at this stage that requires investment, okay? But what we can safely say is that investment is recouped and some by shortening the timelines to candidate delivery. Okay, but it does require investment, and it requires risk taking, and it requires active, advanced decision making. Okay, and with experienced drug hunters. Why are we doing this? Why are we advancing more compounds into downstream assays? Primarily, it's to increase our understanding. It's to increase our learning, and this could be in terms of PK, PD, efficacy, safety, predictivity. It's increasing that translational predictivity that we're so keen to highlight as part of the strategy. Yeah. Clearly, it's to rapidly identify critical blockers. Those critical blockers drive design, but those critical blockers can come quite late in a project, can come quite downstream. So we want to identify them as early as possible, and that will lead to solutions driven by design. Also, the flip side, planning for success, we want to rapidly identify potential candidates because sometimes we don't get those critical blockers. So pushing those compounds through rapidly identifies candidates, and we keep on going, of course. So we do recognize this model requires increased investment, but it is recouped planning for success. And how does it contrast then with an alternative? The best way of describing it, or we name it, is the sort of selection filter approach, yeah, for compounds that fail or pass a set of predetermined criteria. And like you described it as a funnel. So this is where criteria are met, compounds are put through, they pass, tick, and go on to the next phase, yeah. That's a very passive process. It's a very slow and very linear process. And frankly, it minimizes the learning. And what we aim to do is maximize our learning and maximize the information. And in that sense, we overall, we speed up the process. And like I say, that selection filter approach, you can just end up getting nasty surprises late on in discovery that then you need to go back to the start and solve for those particular problems. So our every compound, the candidate learning cascade, it's very iterative, aims to minimize those nasty surprises late in discovery. So that's the best way, Ash, I think, of comparing and contrasting our approach and the every compound and candidate approach that we're putting out there in the community for others to get feedback from versus an alternative.
Okay, thanks, Dermot, Jerome, and Christoph. So that sort of brings to an end the first part, and I'm intrigued to see what questions we've got coming through. So we'll now move to the second part where we engage in Q&A with the audience. So I'll just need a moment to start to gather up the questions. Okay, the questions are coming in, which is good. So we've got a few that I can refer to. So to get us started, so there's a question. So panel, have you already seen the impact of the strategy on current projects? Do you have any examples you can share? Maybe Jerome, do you want to take that one?
Yep, yes, sure. So I will describe two, in fact, projects. We have prepared a slide on this. Next slide. Yeah. So the first one is in the upper part of the slide was a project with timelines to candidate nomination of 2 years. And in fact, during this project, we pursue exactly what we were describing in every compound a candidate, meaning exploring this learning cascade deeply, especially in vivo model, to define those translational thresholds which help us defining the best in vitro threshold to be pursued, to be achieved in vitro, and identify the candidate like this. So it's really efficient from the project start to candidate nomination, only 2 years, with this rapid identification of those thresholds. And the second one is also looking at every compound candidate, but mixed with AI ML tools that we have developed within Evotec, especially because we have started from literature data, and with the help of those specific tools and mixing those tools with our current expertise in medicinal chemistry, we end up with the identification in the hit identification phase with 3 novel, very interesting series for the project, which will then be developed in hit to lead and lead optimization. In less than 18 months, we achieve the identification of these lead compounds by early exploration also of this learning cascades, especially for this project. Higher species like cyno and dogs were very helpful in the identification of the candidate to understand this translational threshold between the human situation and the rodent situation that we have at the very beginning of the project. So also very efficient. In less than 2 years for those 2 projects, we see the impact of this identification of thresholds, progressing more compound into the screening, the learning cascade to achieve very rapidly candidate nomination.
Okay, thank you, Jerome. I'm gonna keep with the sort of projects theme. So a question's come in about sort of timelines and project challenges. So where projects weren't able to be delivered within that optimal time frame, so the average of 2.9, but driving for less than that, what are the sort of project challenges or blockers? Can the panel comment on those when projects perhaps aren't being pursued or prosecuted as rapidly as we would like? Maybe Christoph, you could start the conversation on that one.
Yes, sure. Yeah, so that's a good question indeed. That there is always some there can be hurdles within the project at some point, and what our objective is to try to get over those hurdles. So we've identified that in some cases where we're not able to meet those short timelines, it was linked to many options. That could be first non-optimal communication. You know, we are working as a large team, the partner, Evotec, and so on. So we need also to align on our decisions. So the decision making, which was mentioned earlier, is very important. Also, in some cases, we've observed that a comprehensive in vitro optimization was asked before moving on to the in vivo studies. That was also clearly a blocker for the rapid progression of the project. So not testing at some point the compounds in vivo was clearly a blocker. 100% of the compounds which reach the market are non-perfect tier 1 compounds. So we also need really to test all our learning cascade a bit deeper, and that's also at the heart of the recompound candidate strategy. That's maybe the first part of the answer. I don't know if my colleagues want to add something to what I already mentioned.
So yeah, building on that, Christophe, I mean, we recognize that every project is unique, and the challenges in every drug discovery project, and I've been on many drug discovery projects over the last 3 decades, every one of them has unique challenges. And so trying to describe a strategy that covers all projects and all challenges and all problems within projects is impossible. But what we focused on is tactics and strategies that can be universally applied. So, one way we've described this in the paper is a mindset. And for me, a mindset is focused on the overall problem of reducing clinical attrition, improving translational predictivity, and doing that as quickly as possible with a high quality candidate drug. And so for me, these tactics and mindset is how we approach drug discovery every day, trying to solve those problems and really using the totality of all the capabilities within a drug discovery team to meet that translational predictivity challenge as quickly and as efficiently as possible. That's the way I think about this.
Yeah, just to build on this, efficiency in data generation for the project is also very important to make the right decision at the right time. So that's also something that we have highlighted during our discussion. Be efficient. Operational efficiency is very important to deliver, in fact, the right data at the right moment for the project to make the perfect decision.
Okay, thanks, guys. I've got quite a lot of questions now, which is terrific. So I'm going to try and group them. So maybe a quick one then. So linking into what sits under a project, what types of targets are reflected in your data, in general terms?
So it's highly diverse, in fact. I can take this one. It's highly diverse. In fact, there is kinases, there is GPCR receptors, there is we did not select any one of the other targets. We take whatever project, start with the hit ID phase to the candidate nominations. Recent project, because it's more aligned with what we are doing right now. But that was the only parameter we take into account. Otherwise, from oncology, as I said, CNS also related targets are very diverse in terms of indication of targets, etc.
And John, we could also add that they were different in the term of the starting points, meaning that as you showed the case study, the second case studies was started from literature, but there are also a case where we start with new biology from HTS outcomes. So it can also be, it's a mix similar to what was published in this publication with referring to the 4 years from start of chemistry to chemistry for the pharma industry to PDC for the pharma industry. So that's really also a mix of starting points.
Okay, thanks. Next question. So, we all recognize a candidate on its own doesn't really mean anything, right? Until you're testing it in patients in the clinic, then it's just very much a prototype. Questions come in about, I suppose, tracing success along the path post PDC, and what's the impact of success rates achieving IND, which is that next critical milestone following candidate nomination. I know this is very, it's early strategy, so we may not have a lot of data, but any sort of comments or thoughts on that attrition aspect? Dermot, do you want to come in?
Yeah, we do have some data on that, Ash, for the 15 compounds presented by Jerome, and the majority of those, and again, it's a small data set and it's a small amount of time. These are all candidate drugs that projects been nominated since 2020, so it's in the last 5 years. So clearly, in terms of clinical progression, we don't have a lot of data points. But the vast majority of those 15 compounds are progressing successfully. Indeed, some compounds are now in phase 2. We have compounds in phase 1, and more of the most recent nominations are still in preclinical development. So in terms of quality, based on a small n number of a small amount of time, the quality of the CDs of the candidate drugs seem to be holding up. But again, the proof of the pudding is over time, and this is something that we now have a framework and have a database on, and will continue to as we it's already out of date. We've already nominated more candidate drugs into preclinical development. We will keep monitoring, keep assessing both the speed and the quality, Ash. And the only way to do that really is time. Speed is very trivial to measure. Quality ultimately is looking at approvals of clinically meaningful drugs, and that takes time.
Well, thanks very much. So the audience has perceptibly picked up on the fact that there's several questions on this. So ECC appears to promote the use of increased in vivo studies early on in the project. So how does ECC fit with the emergence of NAMs, new approach methodologies in terms of animal usage, and the push from regulatory bodies to reduce this aspect?
So can I take this, Ash, because I'm passionate about this. So the first thing to say is, personally, but also Evotec as a company, are committed to the 3 R's. Okay, so that's the first thing to say. So to reduce, refine, and replace animal experimentation, in line with the industry, in line with the regulators. That's the first thing to say. What problem are we trying to solve here? The problem that we're trying to solve is improving, or one of the problems anyway, is improving our translational productivity, because we have 90% clinical attrition of small molecule up to candidate drugs. So our translational, by definition, our translational productivity is poor, via whatever process of using in vitro and or in vivo models. So ideally, we want to, of course, improve our translational productivity using human-derived in vitro systems. Of course, that's fundamental. It would only be appropriate to use in vivo animal models where they increase our translational productivity. Okay, so that for me is an absolute fundamental premise of why we use animals in the first place. Obviously, there's a regulatory component, but in drug discovery, where it's a non-regulated environment, then we use animal models by definition to improve our translational productivity in PK, in PD, in efficacy, or in toxicology. And over time, we want to fully replace our in vivo models with in vitro, with human-derived in vitro models. So, and again, that's a push in our organization, just like there is across the industry. So our experience, though, is, and it's absolutely right, we do propose a increased use of both in vitro models, but also there's an element of increased in vivo use early on within a project. What we're doing that for is to maximize the learning, okay? And that's really important. And what that allows us to do, in principle, and again, I can say, we appreciate every project is different, and therefore every project has its own unique challenges. By putting more compounds through downstream assays, be they in vitro or in vivo, early on, we maximize the learning. And our experience is we shorten timelines, we maximize learning, and actually we end up with shorter timelines, but also fewer compounds over time in the totality of the project going in vivo. So actually our experience of applying this in recent times is we don't actually increase the totality of in vivo experiments. And again, we'll examine that over time. But that's my firm expectation that throughout the drug discovery process, we won't end up increasing the amount of in vivo experiments throughout the course of a project by maximizing the learning and increasing the upfront investment in both in vitro preferentially, but also recognizing some upfront investment in profiling compounds in vivo models is necessary.
Thanks, Dermot. So sticking with that in vivo theme and more of a sort of point of detail. So a question's coming asking for clarification if we're doing rat PK on every compound from lead op designs.
Me again, I guess. No, we don't. So we use our tier 1 assay is primary potency, a selection of key ADME in vitro assays, stability, permeability, solubility, etc. And maybe in tier 1, there might be some off-target potentially as well. So all compounds will get put through these sort of high enhanced throughput in vitro assays, measuring potency, ADME, and potentially selectivity as well. And it's only when we have that tier 1 data set that then compounds will be progressed into rodent PK. Rapidly, though, progressed into rodent PK and other parallel in vitro assays, such as secondary, cellular-based on-target assays.
Further build then, and so a question coming in about the feeding of the models by animal data. Does this distort the human-related output from the models? So that connection between animal output and the need to be predicting and translating into humans.
Yes, that's also related to the previous question with rat data. We also try to use those rat data or perhaps higher species to refine the human dose prediction at the end, and to try to like this learn from each and every animal studies and combine those information into comprehensive learning for the human situation at the end. So that's my understanding, but perhaps Dermot or Christopher can say a word on this.
Again, animal data is only used in the context of translational predictivity.
Exactly.
So everything is focused around human relevant parameters, but that's not to say that we can't use animal data to allow us to make that jump, because we have animal in vitro data, animal in vivo data, human in vitro data, and we're trying to make that assessment to human in vivo data. And that in vitro, in vivo, whether it's pharmacodynamics or whether it's a PK endpoint, that provides us learning and information, and it's that totality of that learning and information that allows us to make that translational jump, or it provides us with data that says actually we cannot make that translational jump because there's too much uncertainty. So again, drug discovery provides us opportunities for learning, but also it tells us what we don't know, or can do, tells us what we don't know as well. And we need to therefore go and look at different models or understand the biology better, etc.
Or ask for different in vitro model to try to understand a bit more the in vivo situation. So the blockers are also important, as mentioned by Dermot earlier. The blockers are also part of the in vivo setting, and sometimes you have excretion, you have different mechanism implied into those models. So that's part of the learning.
The one thing I would say, Ash, on this is we don't really have a good understanding typically on the exposure effect relationship, whether that's for pharmacodynamics or efficacy or toxicology. So understanding that, and from a quantitative perspective, is absolutely critical. And that's where sometimes in vivo experiments can be very, very informative, to understand that dynamic exposure effect relationship, understanding target engagements and off-target engagements. And so that kind, but it's in the context of human translational relevance. And we, you know, as an industry, not just us as an industry and as a set of capabilities, we can be that sophisticated to be able to put that animal data in that context. That's what we need to do. And that's what makes every compound candidate successful. It's that combination of in vitro and sometimes where necessary in vivo data.
So I'm just conscious we've got about 4 or 5 minutes left. We've still got loads of questions. So if we can't get through, you know, all today, we'll make sure we get answers sorted. So I'm fully aware of our commitment at Evotec to focus on human dose projections and build those as early as we can. So a question's come in asking about, in medchem design, we typically use Q-cell models. Are you suggesting that medchems can use dose prediction for early compounds as well to complement the next design round? Short answer is, from me, absolutely. Happy for Christoph to build on that.
Yes, definitely, Ash. I think we have a set of models that we can apply anytime in a project. Of course, the domain, the applicability domain is something which is important. But very early on in the project, you can start building on your even very small knowledge to predict your next round of compounds. One example, for instance, in our case, at least at Evotec, where we have the Bayesian optimization, so the active learning, which is running very early in the project. We don't need that many priors to start having our machine learning. So yeah, you don't need to be so advanced in your project to start really leveraging the data you have. First, with the data you generate in the project, but also with the support of the global models that you can generate elsewhere, with the support from the global data. So yeah, my answer is yeah, you can start applying the predictive tools from any time in the project, including very early.
Okay, I'm going to try and do 2 more, just to make sure we fit everything in. So could you please let me know, are there any general workflows for in silico virtual screening processes for lead identification? So I suppose it's just a bit more of a technical question around what you can do in the, you know, the virtual screening area.
Oh, you mean that if the question is about Evotec, we can apply any different approaches in terms of identifying what we call from knowledge-based approaches. Of course, you have the knowledge coming from the literature that you can start from. You can build models from any different sources that can be from the target itself. If you have a PDB structure, you can run some structure-based virtual screenings, but you can also run some ligand-based virtual screening if you know about ligands, natural ligands or synthetic ligands as well. But there are many other approaches that you can think of, like some AI/ML-based approaches where you can build on your molecule growing into the active site. If you know the active site pretty well, you can have some molecular dynamics snapshot that you can screen on. I'm probably missing some different cases, but all the opportunities are possible. You can also run some chemogenomics, starting from a protein from the family of the target of interest and start building on an homology model, or starting from ligands from these other proteins and start building on the selectivity for your own protein. So many different approaches and opportunities to start from. But importantly, it's also very interesting in general, if you want to identify the unexpected, to also run some wet approaches for screening in parallel to the dry approaches. That's at least what I would recommend to do. That's what we recommend to do in general.
Thanks, Chris. So I'm going to come in quickly. So the question may have been more about how can someone maybe get access to general workflows without having the expertise of Evotec, but I would say ask us the question, we'll help you. So that's how I would answer that. Final question, guys, which is a little bit more prophetic and maybe we can't answer, but I think it's quite important and it's a useful comment. So the comment says, while you may gain 6 to 12 months in discovery, clinical development takes years. Ultimately, candidate quality, success in the clinic, etc., trumps speed. and then maybe just a question about how can AI/ML tools help clinical quality. So I think we all recognize this. We're talking about gains to PDC, reducing attrition, but of course we understand that there's many other things that have to go our way afterwards. Any parting comments on that, and then we can probably bring it.
The commentator, the question is absolutely right. You know, quality is king. However, all people working in discovery organizations knows that speed matters as well. You know, so getting that balance, and the every compound candidate strategy, you know, focuses on improving quality, but also speed is important. It's important to all our customers. It's important to most, if not all, drug discovery organizations as well. We're in a competitive game here, so speed is critical. But quality, we cannot sacrifice quality, and improving quality whilst reducing speed is exactly what this strategy aims to achieve.
Thank you. So I'm going to bring this to a close. So Emma, if you could navigate to the slide here. Yeah, perfect. So we're really keen to make sure you've got everything out of this. If you can find out more, you can read the publications. We urge you to do this. There's some handy QR codes that you can use. Please get in touch with us. We're more than happy to try and answer any more of your questions, and we'll do our very best to answer the questions that we haven't got to today. A massive thanks to you all for the engagement. Without that, we haven't really got a session. A massive thanks to my colleagues and panel. Fantastic content, guys. Really comprehensive answers. Hopefully the audience finds it compelling. And of course, thanks to Drug Discovery World for supporting us on this endeavor today. Thanks, everyone.
Thank you.
Thank you.
Bye.
Thanks for listening. A link to today's recording will be available soon at www.ddw-online.com. I'd like to thank Evotec for supporting this webinar, and a big thank you to Ashley, Dermot, Jerome, and Christoph for the discussion. Thanks for listening.